I'll start out with a little bit of kind of an overview of what it means to make drugs in a biotech company. Amgen started out as just biotech, and I didn't start out at the beginning, but I started out before we had a product. So back then everything was R&D because we didn't even know if you could make a protein and use it as a drug. The only protein that had been used as drugs was insulin up to that point, which was very different. So that part was earlier, but as you start to have products and things that you're putting in humans, the process development part actually becomes pretty important. And I have a feeling that probably a lot of you don't know what that means. I don't think I would have known what it meant if I hadn't gone to industry. And especially that even though we're kind of the later half of making the drugs, there's still a lot of research there. And so that's partly what I'll talk about. And then a little bit about the tech dev programs that we have. And then I heard that you wanted to know a little bit about career path too, so we can talk about that at the end. Though I'm not sure if that made me kind of think about why I ended up where I did, and I think part of it is just being really stubborn, which I'm not sure is necessarily good career advice, but the way it is. So please interrupt, ask questions. Do anything like that you want to. Yeah. So let's see. Does this advance it? Oh, wrong one. I think I'd be able to figure it out, huh? Okay. Never mind. I forgot to ask you how to how to make the slides go. Okay, there we go. Oh, so I will say one thing. So my title says scientific in front of executive director. So that says that I chose to take the scientific career path. At Amgen, at Amgen, Genentech, and definitely MedImmune, and many of the companies that started out as biotech companies, there is a science path. And what that means is that I chose, and in my case actively chose and resisted, going into management as a way of having influence over the strategy and what happened. And instead really chose to kind of concentrate on the science and science strategy. And so there are jobs for people who are really more interested in that. And actually at Amgen, if you have a really deep expertise in a particular scientific area, and that's really where you're interested in staying, you can do your whole career and stay there. It probably means you'll never be a vice president, but I've seen what vice presidents do, and I have no desire to ever do that. So, so there is those kind of options as well. So, when we start out, we do have to think about things like cost. And what you see up here is in 2006. It cost about $500 million to $2 billion and took at least 15 years to produce a successful drug, especially a protein-based drug. The result of that is that we can make medicines that can cure people that are ridiculously expensive. And some of you might have heard about the hepatitis C drug that can actually cure people from hepatitis C, but they did a calculation and figured out that in California alone, if the state paid for that drug for the people who need it, like seriously need it or will die from complications from hep C, it would be billions and billions of dollars and the state would go broke. So that's not a tenable future. We can't keep making things that might work, but that only really rich people can buy, and that are going to bankrupt governments. And especially as kind of the model for how you pay and how medicine happens is changing across the world. And personally, I didn't go into this because I wanted to just cure really rich people in the U.S., right? I went into this because I'd like to make medicines that can make people all across the world better. So really, the next 5 years, 10 years from now on, are about learning how to do what we do in a way that's much cheaper, get better at taking the right risks so that we can do things quickly and get it into people. Because one of the things we've learned is that no matter what model system we have, nothing is humans. And so, even if we try to have a lot of good preclinical models, either in vitro or even in vivo, that's why you still see things get to clinical trial and then that's where they fail because something unexpected happens. So we have to figure out how to make something safe enough to get it into humans and then find out if it works. So it's kind of a different paradigm. And because of that, actually, Amgen just underwent a pretty big reorganization to try and think about how can we do this better. And it takes knowledge, understanding, and definitely persistence. And then the continuous learning and improvement is what I'm talking about. Now that we know how to make proteins that can be stable enough to be used as drugs, have the shelf life that we need to start thinking, when should it be a small molecule? When should it be a protein? When should it be a hybrid? What can we do? And what can we learn from both the small and large molecule world, which used to be very separate, and we didn't talk to each other. And that's not true anymore either. So one of the big things is, at least at Amgen, we only want to do things that are a significant medical need. So early on in my career there, I actually worked on a protein called KGF, which is what makes your skin cells grow. It turns out that actually would probably be a cure for certain types of baldness. We didn't pursue that because that's not what we're interested in putting our money in. That's not what we're developing drugs for. When I started, we actually weren't only doing human pharmaceutics. We were doing human pharmaceutics, animal pharmaceutics, and chemicals. So one of my first projects was I worked on 3 different human pharmaceuticals, and then I worked on cetylysin, which is the protease that they put in detergents to clean up the stains. We engineered a molecule that worked very well, but that's much less fulfilling to know that maybe I made your shirts whiter. It's not the same thing as knowing that I actually helped people get through chemotherapy quicker, And then the other thing is, can it be successfully treated? And what are the risks and benefits of treatment? So we always want to make sure that we're not doing harm, that we're not curing one thing and making things worse. That's just kind of common sense. I put TNF here, though, as an example. That's tumor necrosis factor. So when I first started, people knew that there was a protein called TNF that you injected it into tumors or mice with tumors, and the tumors shrunk. And that's absolutely true. We also knew that there was a protein that people were calling cachectin that when you gave it to animals caused their immune system to totally get excited, cause all sorts of reactions, really high fevers, and the mice would die. It turns out that's the same protein. So now it's very well known that TNF is one of the main proteins in the immune system that turns on the cytokine release and our response to drugs. This is a good example of, until, in this case, once we got it into animals, we actually eventually figured it out. Because if you start out with just a gene sequence or just a protein sequence, a lot of times we don't know what they're doing. One of the things that's changing this paradigm are efforts like Decode, which is a company in Iceland that Amgen has now, but the most important thing is what they have is a gene code from everybody in Iceland. It's a small country, so there's like 300,000 people, but people have actually given the government the okay to actually have their gene sequences, and people there can trace their ancestry back like 1,000 years. So there's much more variability. The other interesting thing with Iceland is it's about a 50-50 mix genetically of Vikings and the Celtic people on the coast of England that the Vikings encountered on their way to Iceland, actually. So it's got some genetic diversity. And they can do studies like when you've seen the Amish trees where people follow the genetics, but much more diverse. And so that's one of the things that people are starting to think of now. Instead of putting things in the clinic or putting it in animals, if you have a gene or a protein that you think does something, one of the things you can do is go back and actually look at the genetics. And is there a disease that's associated with that gene if you follow people's family trees? So those are the types of things we can do now with big data that will really help us, I think, make better medicines faster and targeted that actually work. Okay, so first you find the target, first you find the disease, then you find the target, which is what I was just talking about. So you can see here that many of the genes, we still don't know what they do or if they can really be targets. The validating as a mediator of a disease is what I was just talking about with, that maybe the use of big data and genetic profiles from people can help with this. But you have to be careful with that, right? Because all of us have all sorts of mutations, so you have to make sure that you're not putting emphasis on things that might not matter. And so one of the easiest things to go after is signaling molecules or receptors or enzymes. Once you figure out what the target is, that's when then we can think about what kind of molecule will actually work. And in the past it was just small molecules, and then after biotech started, it was small molecules and proteins. Now there's RNA, there's cells, there's those SNPs now where maybe we can go to DNA. So I really feel like the next 5-10 years are going to be as exciting as the first 5 years when I was at Amgen, when we were really finding out if biotech could work. I think it's really a revolution. And we can try and really target the disease, choose what we want to target the disease that we want in the best way. And this is just to help people kind of get things in perspective. Aspirin and we call them small molecules, they're really organic chemicals, are very small. You can define them and know everything about them. And then we're talking about making things like Enbrel, which is huge. And now this is, antibodies are bigger than that. We have things that are 2 antibodies together, 4 antibodies together. We're talking about making molecules that are hundreds of thousands of Daltons and having that 3D structure right. So actually the making them is really challenging, and that's where the process development organization comes in. So we pick which one to go if we're being rational and not like holding our particular modality precious because we like them. It used to be the large molecule and small molecule chemists would get into big arguments with each other about which one was better, and really that's absurd. They're better depending on what they want. Small molecules are really good if there's a small binding pocket that they can fit in and cause all the reactions to happen. But when you have things like surface cells and you need to pull cells together, or you need to activate receptors on the cell. If it's a more complicated system that you're trying to turn on, that's when you need proteins. And that's partly why antibodies ended up being such a promising therapeutic, is because they're big and you can put targets and make them link together the things that you want linked together. But as of yet, there's no large molecule that's really been consistently able to access the central nervous system, and some small molecules can do that. That's changing right now as people really work on different delivery systems. And I'll talk about delivery systems towards the end. And then one thing to remember is with small molecules, you can take them orally, and that's a very nice way of taking them, but they have a very short half-life. If you're taking aspirin or acetaminophen, you have to take them every so many hours. Even really good small molecule drugs you have to take at least once or twice a day. With the proteins, they have a much better half-life, and so maybe you can get an injection once a month, in some cases a couple times, and then you're done with it forever. So that they're better from the dosing frequency standpoint. And then another difference, but for proteins, if you take them orally, we eat proteins all the time. If I give you a protein drug and you eat it, unless we do something special to make it not get digested, your body will just think I gave you some more, nitrogen source for energy, and it won't have it do anything special. And then another difference is, so DS stands for drug substance. When you're making a drug, the first step is to make the drug substance, and then what you do later turns it into the drug product that's actually what people are given. For large molecules, the drug substance is often made by a series of chromatography steps, and say in the case of proteins that are based on antibodies, those steps can be very, very similar. So the drug substance part, the purification part of large molecules, is actually, in some ways, the easiest part. My colleagues who do purification don't like it when I say that, but that's where I started when I started at Amgen. So I think I get away with it. The really difficult part is making the cells make the protein that you want. And then sometimes you'll have molecules that aren't very stable, and getting them through the purification is tough. But for us, that tends to be kind of the part that we can platform. I was talking before about making things cheap. One of the best ways to make things cheap is to be able to do a platform things where you repeat the same methods and you can learn from one molecule and apply it to the next. So that's a really important thing. For small molecules, the drug substance is actually the tricky part, because that's a series of unique chemical reactions. You're synthesizing an organic molecule. And so that's another difference between the two. I know I talk really fast, so I should have mentioned to you guys, if I'm going too fast, wave your arms and I'll try and slow down. So then this is just some other differences. And I think, I sent the slides, so this was more I put in here in case people were interested and wanted to look at it later. A couple of things here are let's see if this part works. Yes. So, for characterization, for small molecules, you synthesized it. You know exactly what that is. And it's very easy to see what the three-dimensional structure looks like, too. For large molecules, they're much more complex, and it's actually very difficult to fully characterize them, partly because proteins are dynamic molecules. So even if you see the X-ray crystal structure, that doesn't necessarily tell you anything about how that molecule is in vivo when there's other proteins around. It might change shape when it's binding to what it needs to bind to, and that flexibility to change shape is absolutely crucial. I can think of at least 5 instances when we've engineered a protein drug to make it more stable and managed to make it so stable that it could no longer function. So you have to always remember that about proteins too. They're kind of living dynamic molecules. So we want it to be stable enough that it'll last in your body to give you the benefits for as long as possible. And we also have to have it be able to exist in the pharmacy for 1 year or 2 years, but we also want it to be flexible enough to do its job. And if you think about that challenge, in our bodies, proteins evolved to do their job, get turned over and disappear. The last thing you want in your body is the protein EPO, which makes your body make red blood cells, existing forever and ever, and you having the wrong amount of red blood cells. You have to be able to control that. So when we talk about making proteins as drugs, we're actually going against thousands of years of evolution to try and make something that'll actually be stable and keep doing its job for as long as we want it to. And that's one of the things that when I talked about continuous improvement, we've learned more and more about how to engineer proteins to make them more stable, but try not to screw them up too much. Then whenever you think something happens that totally surprises you, so that keeps it fun. And so because of the characterization thing, comparability is really easy here. Here we have to do a lot of comparability tests to show that when we vary from lot to lot to lot, that it's always going to be exactly the same. Okay. So this is we're going to talk, since my background is in large molecules, and I think that's what people are probably the most interested in. That could just be because that's what I'm the most interested in, but anyway, I'm going to talk mostly about that, but I have some slides showing the other one too. So this is just from the discovery research to final molecule selection. And so all this part, the target, figuring out molecules that might work, screening things for activity, going through a bunch of engineering to pick the right one, down to purifying enough to start doing analysis to see if it actually has the biology that you want. All of this part is done in the Discovery Research Organization, or in our R&D organization. Once it gets down to the top, say, 5 to 10 molecules, then actually the department I'm in, process development, takes over. And so we start here with cell line generation to the final molecule selection. So this part is jointly owned by our R&D department and our PD department. And that's because we still want a molecule that has the right activity, that has the right safety profile, and the people who think about that and can do the assays, do the animal studies, even set up the clinical studies, they're in the R&D organization. But if you have a wonderful molecule that can do everything you want to, and it's impossible to make it at a commercial scale, it will never be a product. Or if you have a wonderful molecule that will do everything you want to, and its half-life is a day, that will never be a product. So we also want to make sure that we're selecting a molecule that we think we can actually turn into a real drug. And that's where the expertise from the PD organization takes over. Where this actually happened has changed multiple times. So I've actually been in R&D and in PD and back in R&D and back in PD, because this is where my functional responsibilities were for a long time. You can probably tell from what I've talked about so far, I get really excited about protein structure function. And so that's where I am. And so we call this candidate selection or molecule assessment. And we call it molecule assessment to kind of make people realize it includes the activity, but it also includes the ability to make it. And that's because before this part got added, people were making decisions based on activity differences of, say, 20 or 30% in a bioassay where the variability could be 50%. So you can't really differentiate. And yet they would pick one, and we had a couple examples where the one that got picked turned out not to be the one that had the stability or the other appropriate properties. And we had to go back and look at one of the other ones of the 5. So that's why we do this jointly together. So the commercial product part means, can you express it and purify it to get yields that are okay? And when you express it, can the cell make it consistently the way you want? For proteins that have sugars on them, that sugar profile has to be the same. In the case of antibodies, the sugar on the protein actually determines how fast your body turns it over. And also, if it activates the immune cells through the complement pathway and some other pathways that our bodies have. And you might want it to turn those on. If it's something that you're giving to people for cancer, you absolutely want those ADCC pathways turned on because that's what's going to kill the tumor cells. If it's something that I want to give you because you've got rheumatoid arthritis or some other chronic disease, I absolutely don't want those turned on. And so the sugars are actually very important. The ability to have the cells make it consistently is absolutely crucial. And then the purification yield. We want to know that it doesn't have a lot of aggregation. Protein aggregates not only cut down on the yield, but they can be efficacy or even more, have more potential to be a safety issue. We put viscosity here. It might seem funny to you that that would matter, but it turns out that in the case of antibodies where you need a high concentration of the drug, if you give somebody something that's too viscous, you actually can't inject it. There's a limit to the amount of pressure that humans can tolerate in their skin when people are giving you an injection, and really viscous solutions are incredibly painful. And also the devices that we use actually sometimes can't even make them go through. So it turns out that one of the things we have to think about is when you put the protein at high concentrations in solution, is it very viscous, very thick, or not? And that depends on the proteins interacting with each other and kind of forming networks in solution. So that's one of the things that I think people have just started to understand in the last something like 5 or 10 years that we never would have thought of before. And then I mentioned the stability before, that we have to be able to put it. And then formulation is very important. The formulation is the final buffer or solution that the drug will go in. Or if it's going to be a solid, it's whatever makes the final drug product what we're giving you. But for solutions, and most large molecules are in solution, it has to be something that we can tolerate. So it has to be isotonic. I don't know if any of you have had the MMR vaccine as an adult. It stings like crazy. I had to have one, and I realized no wonder the babies cried. It's because of the formulation it's in. It's in citrate, and citrate is not a good thing for humans are sensitive to that. So you want it to be in a formulation that's tolerable, but also that keeps the protein happy. And keeps it from aggregating and all those other things. So figuring out the right formulation, there's a lot of interesting solution chemistry that the formulation people know. So these are the things we test it for. And then once you have the final molecule, then you go to the whole process that has to happen to end up making something actually through all the clinical trials and to launch. So process development is the group that picks the final cell line. I'll talk about the cell banks in a second. Makes the material, drug substance, drug product, and all the analytical testing to demonstrate that we can make something consistently enough that it can be a protein. FIH means first in human. I don't know why, but in industry, people love acronyms. There's so many acronyms at Amgen that there's even some where the same initials can mean more than one thing. So finally, on the company website, there's actually a page that's acronyms to help people know what you're talking about. It's ridiculous. But anyway, so FIH really means this is the getting it into the phase 1 trials. So making all the material for this. And then the toxicology studies and the phase 1 and phase 2 trials. The trials themselves are done by our clinical trial group. But the material that's used is made by process development. The analytics that are done to demonstrate that it's okay are developed by process development. Then you transfer the analytical testing to the quality group, and they're the ones that do the routine lot release once you're manufacturing a product all the time. We don't do that anymore. We go back and start developing things for new proteins. And let's see what else is important. The top formulation candidates, I mentioned that this is a lot of work. Often what happens is to get it into phase 1s, you'll just kind of pick a formulation that's good enough and then do a lot of work to get it into the formulation that's exactly what you need for that drug. This also depends on the device. If it's going in a syringe, in a vial, now there's really fancy devices that people can wear on their bodies that'll inject them later, that'll inject them slowly. Depending on what device your drug has to go in, that will determine what solution you can put it in. And it'll also sometimes determine what protein you can use, because the protein has to be compatible to all the surfaces it's going to touch. We had a problem about 10 years ago or so where it turns out in the syringes, when they make the little hole, they stick tungsten pins in there. And they were doing it at such a high temperature that some of the tungsten was sublimating and sticking on the inside of the syringe. And all of a sudden, we had proteins that were precipitating only in that part of the syringe. It took us like almost a year to figure out that it was from tungsten. It was a really fun problem-solving thing, but it meant that nobody could figure out what to do with those syringes, and we had to work with the people who we buy the syringes from to figure out the process. Never, and they were using tungsten because tungsten is an inert metal, which it is, but it turns out that tungsten oxide, a certain kind of it, with proteins makes these nice colloids, and then they aggregate. So those are the kind of things that process development does. We figure out the root cause for when things go wrong, like detectives too. But it means you also have to understand your device. So we have a whole group that's device engineers. And now, as we're kind of moving into the new world of trying to make things that are directly targeted for the disease and the person, a lot of times that involves the device right from the beginning. What could make this be usable for that patient population? And then I did put here, and then analytical support. So once you hit launch, this is when we all get excited and celebrate because it means that we have a real product. And so you can see from these two slides I showed you, that's what takes 15 years or whatever. And depending on the clinical trial, sometimes the clinical trial can take more than a year depending on what your outcomes are too. So that can add to it. But even here, we're not done. Once you launch something, then it goes to the commercial manufacturing group. We have manufacturing sites all around the world now. They have to make that drug the same way every single time. And it has to be tested to be demonstrated that it's the same every single time. And when you're going through the lot release testing, if you see things that you didn't see before, you have to stop and understand why. And that a lot of things lately have happened from devices. So some of you might have read about a bunch of different companies had to have recalls of lots that were in glass vials because there were little glass lamellae forming. And it turned out there was probably one very subtle change at the manufacturing company that made the glass vials. And that, in combination with particular formulations, could make little teeny pieces of glass flake off with time. Again, something that none of us would have thought about, but you have to stop and solve that. And then if there's really a glass in a lot of your proteins, you have to recall that and put it in a different vial. So we're constantly learning, and in PD we're responsible for those kinds of root cause analyses. I just wanted to mention cell banking because I said how important it is to have the cells making the same thing all the time. This is actually done because they're clones or very close to clones. So really it's the same cell over and over and over and over again. You make a once you have your final cell line developed, you make a master cell bank and you keep that. So then you can make the same thing. And then this is just to show you, this is a typical purification process from beginning to end. So I said that it was the easy part. What I meant was some of this part you can use the same column, say, over and over again, but you can see still the whole process, it's not actually easy. All right. And this is just a picture, then, of what some of the facilities look like. And the size, so these are columns. If you guys have run columns in labs, they're usually pretty small in glass, right? The biggest one I ran in grad school, I had to run a 6-foot tall column, but it was still 6 foot in glass, right? These guys, so this is actually a chromatography column. It's just, really big. You can imagine the number of theoretical plates that are in there. And then when you get up to commercial production, it might be even bigger. Now, this is how things were. We're all moving towards actually growing the cells in plastic bags. So the bags will be inside the stainless steel, but everything's movable now. So it used to be if you went into a protein production plant, it would look like a brewery. To me, the first time I went into a brewery, I was like, oh, home. But everything would be hardwired in. There'd be this column, and it would be plugged into the water and the electrical, and everything had to stay where it is. Now, if you walk into some of these factories, in between processes, it just looks like an open warehouse with a bunch of columns with the utilities there, and you wheel in what you need depending on what process you're doing, and then you can wheel it out. It's pretty cool. And it's one of the ways that makes things cheaper because it's disposable and you can reuse things. And you don't have to build a new manufacturing plant every time you have a new product. So this is just reminding us that the drug substance involves product expression, purification, storage, and controls. And then the product development is really all the final forms, including the packaging. We have packaging engineers. It turns out that the packaging and labeling is like a really huge deal. The FDA really cares about that. So for small molecules, then it's a little bit different. I mentioned that you have raw materials, and then you go through your synthesis eventually to get what you want. And for them, a lot of the issue is what are the raw materials and the API starting materials, and they have to come from things that are approved to be used for humans. So there's a lot of, this part's really crucial for that part. But then again, in the end, they have the drug substance and then the release. And we're all controlled by the GMP regulations for that. And there's, in many ways, there's very similar things that you have to think about. Cost, the yield. In this case, they'll develop the synthesis that, the simpler the synthesis is, the less it's wasting, and if it's environmentally acceptable. No matter what we're doing, we always try and think about making things acceptable, and nowadays especially to recycling as much of the water and everything like that we can. And then this is their drug substance development activities. So again, it's very similar. You want to get things into phase 1, and then once you know that it's working, you'll go ahead and optimize and really get the things that you want. I'm thinking, yeah, so it's this. So see, no matter whether it's large or small, it's the speed to the clinic to testing it first that really helps us out. And then this is kind of what a chemical lab facility looks like. Honestly, can you see these are the big? To me, actually, some of this really looks like a brewery too. And then this is for making the drug product. The part where small molecules and large molecules are really different is in the drug product, because for them it's very often solid and tablets. And so I mentioned how for proteins the formulation and the devices can be really complicated. For the drug substance, actually, once you have what you know what you're doing and you're making it into a tablet, that part's often the same. So that's what they can platform. And nowadays, actually, people are even experimenting with 3D printing of tablets, which is really intriguing. So. And this is showing what it really looks like when you make the tablets. You have to control things like the humidity, and exactly what the crystals look like that are forming the tablet. There's still a lot of quality control, but it's really different physical parameters than what we think about for proteins. All right. Okay. So that's the what PD looks like part. Did you guys have any questions before we go into the tech dev? So that either means that I made everything really clear, or you're totally confused and just wish I'd continue. So I wanted to start out with that front part to show you that there's lots of places where we have to do research, that we really still don't understand things. So one of the things we're doing is really driving things with the target product profile. So that means we know that there's a particular type of cancer, so we can define what's the target, what's the patient population, what would be the optimum way to give this patient population the drug, and what would be the optimum characteristics that we want, and what are things that don't matter. And once we have that, then it used to be that we said process defines the product. So when you filed with the FDA or any of the regulatory agencies, what they cared about was, can you do exactly the same thing every single time? And if you wanted to switch manufacturing from one building to another, even on the same campus, you would have to do a lot of comparability studies to show them that what you made in building 1 and building 2 was exactly the same, because it was the process that was important. Okay, that's really secondary, right? What we care about isn't actually if the process is the same. What we care about is the product that you're getting the same. So now we've switched to the product defines the process. This is called quality by design, and it just means you want to understand the parameters within which your process can vary and be the same. So say pH. I want to know when I'm growing my cells, what's the pH range at which they'll still make exactly the same protein. And when I'm running a column, I want to know what's the ionic strength and the pH and the variations in buffer components that will give me the same purity of that protein when I get it out. And once I know that design space, then I can have things vary within it, and I can know I'm getting the same product. So then it shouldn't matter if I'm making it in the US or if I'm making it down in Puerto Rico, or if I'm making it in Holland. If I'm using the same process parameters, I'm going to get the same product. And then even more than that, we want to understand which attributes matter and which modifications have little impact. So there's a lot of data that suggests that for antibodies, methionine oxidation in the Fc region does not impact safety or efficacy. And yet still, when we make a new antibody, we do a lot of testing to show that that's not being modified or that we understand that it's being modified. And if we have a lot that has a slightly different amount of oxidation, we'll still go back and do those tests. That doesn't make any sense if we're trying to make things safe. I would rather pay attention to the changes that I know are having an impact. And so this has kind of been a paradigm shift. I think other industries had this philosophy all the time, but because we're making things that are going to humans, it took getting a lot of experience with the protein drugs themselves before the regulatory agencies and the industry was comfortable enough to say this is what we should do. In order to move to this type of an environment, we have to understand the critical quality attributes. We also want to have high throughput methods. We want to have automated methods. And we'd love to move to the predictive space where we have models. So I can look at the amino acid sequence of a protein and figure out what needs to be changed to affect the viscosity. We'd like to do as much as possible in silico as we can also. And then we're trying to develop continuous processing and flexible manufacturing. That means that you put the protein, the cell makes it, it goes through the purification process, through formulation, and comes out done continuously instead of in discrete steps. So these are all things we want to do. In order to get here, we have to do a lot of research. And so what we do is we have our own technology development. And that's my job is kind of coordinating that effort for process development. I did this for the smaller group I was part of before. And I think that I really like cross-functional things. I love connecting people with different backgrounds to make us come to a whole. So some of my favorite things I worked on was like what I talked about already with the tungsten and stuff like that, where it took a bunch of different expertise to understand it. And that's really what you need for efficient tech dev. And I put this little thing up here. We'll talk about innovation all the time, and that's like a key word everywhere now, right? Everybody's always talking about innovation. For us, it means everything from ideation to implementation. But if you can't actually implement something, then it's more on the invention stage. But to me, it's not innovation. True innovation includes actually that it's something that people use. And we want to make sure that we have this balanced portfolio. It's really easy to think of technology development things that are just a step away from how we've always done things. And especially in industry, if people start telling you you have to put a dollar cost on what your innovation is going to save the company, you always end up here because you can only put dollar costs on things that you know how they'll work. We want to make sure that we have some things that are really revolutionary. These are the ones where I can't tell you how much money it'll save because I can't tell you if it'll even work, but I can tell you what I think it will contribute to the business or to science and why we should do it. Then we want to make sure we're doing some things here too. But eventually these projects need to move into being implemented, or this is the area where we might, instead of doing it ourselves, have an external collaboration and fund a university lab to work on that. And the reason we do that is because a lot of the problems that we have, we still need research done, but they're not things that professors can actually get grants through for the NIH and things like that. And so it's this type of academic collaboration between industry and academia has become much more common. The longer I'm at Amgen, the more common it is. Though, because Amgen and Genentech were both founded out of universities, they've always had some of them. So for us, we have what we call strategic imperatives, and which is, because we are a company, you can't just work on anything you want to. It has to be something that's going to deliver to what we need as a business. Honestly, I think in academia now that's often true too. It's you can't just work on anything. It has to tie to something. But for us, that's very true. So our strategic imperatives are things that we think we need to work on in 5 to 10 years from now. And then we have all these other programs, but an important part of it is the postdoc program. And then, like I mentioned, external collaborations. So this is just how the imperatives work. They're like guardrails. This should work for people that are in California, right? So you want new ideas to come on. If it's something that we're just working on one person at a time, it'll be like what I call the slow lane. But then if we think it's something that's really important, could be really impactful, we'll put together a cross-functional team and we'll resource them. Like say you have a dedicated amount of time and money to spend on this. And then these off ramps can often be things that we'll have with academic collaborations as well. So we have the postdocs. We've had postdocs in PNPD for 8 years. Now with the reorg, they're kind of launching a bigger postdoc program. So there's going to be postdocs in R&D and in PD, so across the organization. So this is kind of how we prioritize them. In PD, it has to deliver to the tech imperatives. It has to be beyond what we can normally do. A postdoc can't just be, instead of another scientist, just a pair of hands. It has to be more research. There has to be the ability to publish and to do presentations, at least on part. And that's because most people that are postdocs are not going to necessarily get a job at Amgen, right? And if it's a true postdoc experience, you've got to be able to then go and get another job. And that hasn't been a problem for our postdocs. Because of that, we also make sure that the mentors are people that have a demonstrated publication record that we think will be good. If we have a junior scientist who has an awesome idea, what we'll do is pair them up with a more senior person, and the postdoc will kind of work with both of them. In general, the postdocs have ended up being kind of a nucleus, and a whole one of these cross-functional teams will form around the postdoc, because you're usually working on something that we've decided we want to resource. And it's really awesome for people to have someone who's doing 100% of their time on tech dev. It's easy to feed them ideas and then support them by doing some pieces of the research that you can if it's interesting. And the best, most successful postdocs have been like that. So these are some of the things that people have worked on in the past 8 years. I thought you might be interested. A lot around the cell metabolomics. I talked about how important it is to understand the cells. We have our factories, our cells. We really need to understand how to engineer those cells to make what we want. That means we have to understand their metabolism. The characterization of protein aggregates and immunogenicity was actually my postdoc, so I know the most about that. And I looked back at the statistics. About 30% of the people who were postdocs became full-time Amgen employees. Sometimes in the area they postdoced in, sometimes in other areas. Most of the others went on to work in industry. I think there's only one who went into an academic job. I imagine that's because if you pick a postdoc in an industry, it's partly because you want to see if that's where you want to stay. And all of them have had external publications or presentations, except one person I can think of, and her career took a different path. And then we have the external collaborations, as I mentioned, which can be 3 different kinds here. Again, each of these are expected to result in joint publications. It's really easy for us to give people protein. A lot of times we have I said we have the 5 candidates and we pick one. That means we have protein left from 4 that are related structurally but didn't succeed. So, those are actually often very good archives to do research on. And so it's good to be able to do that with people. And again, the academic collaborations, we kind of have criteria for them. In this case, we will use an academic collaboration to do something that we could do in-house, but we don't have the FTE. So it's different than postdocs in that respect. And sometimes, honestly, it can just be because we want to establish better ties with some of the universities. So part of the reorg that Amgen just did was to close down some sites and say we're really going to concentrate in San Francisco, Thousand Oaks, and Boston, in Cambridge, right by MIT. So that means that we're actively going to try and have better ties with the academic institutions by San Francisco, in Southern California, and in Boston. And so that's kind of changing some of the academic collaborations too, as we try and kind of build better relationships. And that's partly why Donna and Bill came and talked to you guys initially. We're really interested in establishing ties with the university that's close to the site where we're at, for the postdocs at both places to communicate. Now, my group, I should say PD, actually doesn't have anybody in San Francisco. We have a lot of people in Thousand Oaks and in Cambridge. That's just how things split up. So process development is a critical function at Amgen. It includes lots of modalities, process, product, analytical, and formulation development, and I should have put devices up there. So you can imagine our staff have a wide range of scientific expertise, which makes it really fun. There's always somebody if you have a question, you can always find somebody who should be able to answer that because that's their area. And we do everything collaboratively. you have probably heard people talk about like a matrixed organization or working in teams. We have the Amgen values. The first one is be science-based. The second one is collaborate and work in teams. The next one is trust and respect each other. Then it goes into other more business things. But that's because that's really how we function. And for tech dev, that's important, is the being able to work cross-functionally. And then we actually have a technology development program. I think that surprises people sometimes to hear that in industry we actually put that. And so I'm at the executive director level, which is I've been there for a long time. It means they pay me a lot of money to spend my time just thinking about how to do this, and the company wouldn't do that if they didn't think it was important. So, do you have any questions you want to ask about the postdoc program or anything? And then.
Is it aimed more at graduate students going into their first postdoc, or if you already have several years of postdoc, say for Stanford, what position would you be expected to go into?
So if you want to try a postdoc because you're still interested in seeing if industry will work or not, that's fine. And we've had postdocs from straight out of grad school to it was their third postdoc. All postdocs are compensated the same. That's just how they do it, to make it equivalent. So the amount of experience you've had before you postdoc at Amgen, you'd still, as a postdoc, it's the same. Once you get a job, if you were to get a job, they might factor that in. But we don't. We've had all of them. I think it depends a lot on what people want out of their career and why they want to try an industrial postdoc.
So it's seen as a temporary position.
So postdocs, if you're hired, it's expected that you'll stay at least 2 years, and it's expected that Amgen will have you stay 2 years. But really, we assume it'll be 3. So most people stay for 3 years because, a lot of times it takes 2 years to get stuff, and the third year is when you can really get good results. We've had some people stay for up to 4, but no longer than 4. And then after that, you can't get another postdoc position at Amgen. You would have to either find a position or go someplace else. So it's treated as a true postdoc position. It's not a stepping stone to a full-time position. And that's why I thought it was important to mention. 30% get jobs, so that's better than nothing. I think Genentech likes to hire none of their postdocs. Or some programs will say that, but it's also not a guarantee. But I thought it was interesting that most people who had postdoc did go on to having a postdoc, position.
A lot of us here are postdoc already, so the next step will be looking for scientist positions in industry. Can you comment on what kind of positions openings you have with PD at Amgen?
So right now there's positions. It's a good time, actually, because after the reorg and restructuring and stuff, there's a lot of hiring happening, and most of the hiring is happening in San Francisco or in the Cambridge area, but some in Thousand Oaks, across the board. And typically, for a scientist level, you want someone who has an expertise that would match up with what you want, but you also want people who maybe have a broader background, can bring something you don't know. So that's no matter of trying to see what interests you. So I know I can see I've talked for a long time, but they're asked about career paths. So for me, like I said, I think there's 3 things that define what I did in my life. One is being stubborn, and kind of when I thought that something was really right for me, doing it no matter what people told me. At my age, being a girl that chose to take, chemistry, I was in the honors chem program at Michigan. There were 2 of us and 50 guys, right? So I had to be kind of stubborn, or I wouldn't have even probably done that. When I had my son, I was the second female scientist at Amgen to get pregnant. So I'm used to, kind of being one of the few, and I think you have to be stubborn. I'm a redhead, so I got teased my whole life, so I think that made me kind of not care what other people thought from the beginning. But then there's also listening to people at the right time, right? So sometimes, you need to listen to people's advice and then decide if it fits with you or not. Nowadays, and I think this is going to be especially important in the future, people who can work at the interfaces of fields, who really have a broader background and really like collaborating and thinking outside of the box, that's really important. And that's kind of where I've lived my whole life is at the interfaces between different things. And then the other thing I would say is, if somebody offers you an opportunity to do something and it scares the heck out of you, you absolutely have to say yes. those are the best things, you know. So like twice that happened to me. So I started out at Michigan. I ended up at UCLA actually because my husband went to graduate school at Caltech. I was sure I was going to hate California. I thought there were no trees in California. I don't know why, but you know, it's true. It's not as green as Michigan. But got married in December. I only applied to UCLA and Berkeley actually for graduate school, and that was because I wanted to do proteins. When I got my PhD at UCLA, I was the only graduate student doing proteins, not just in our small department, but in all the biological chemistry departments, because that's when molecular biology was a new, really big deal. That's when they were just building the Molecular Biology Institute at UCLA, and that's where the chemistry people had to go. I was in a very tiny department that's actually in the medical school. And I was lucky enough that my professor wanted someone to work on proteins, and he was actually a lipid chemist. So we kind of learned the field together, which was really cool. And the protein that I ended up working on is a cytochrome P450 that it turns out is a bacterial model for the mammalian ones that are induced by aryl hydrocarbons and get rid of drugs but also cause some things to become cancerous in humans. And the genetics are a model for that. So actually, the first paper I ever published is my most cited paper. So my career's gone downhill ever since, you know. And then when I went to, and then I did a weird thing. I actually postdoced in the same lab because I had figured out how to induce the protein, how to purify the protein. And I decided I really didn't want somebody else to come in and spend the last 2 years getting all the fun characterization data. And then when I decided to go to Amgen, actually I had every single person who had been on my committee sit me down and tell me that I was committing career suicide because you weren't going to be able to do science and industry. And they had a tenure track position that they wanted me to take. And I had always thought I was going to be a professor, right? My whole life I thought I was going to be a professor. But I really liked the idea of applying what I'd learned. So if I had stayed at UCLA, I would be the world's authority on that P450, right? That's what I would have worked on my whole career. And I would have known that better than anybody else in the world. I can't tell you I know anything better than anybody else in the world. But by going to Amgen, I've touched thousands of proteins. So I think I have a broader vision. And to me, sometimes that's part of the difference. You know, the obvious difference is in biotech, what you're doing is applied. You're making something that's going to fix people. But a lot of academic research, you know, can be applied and in the long run does things like that too, right? But the other thing is that in industry, you're going to be required to think broader. You know, you're not going to be able to become the world's expert in one thing. But if you like different pieces of information and pulling it together and coming to conclusions, then I think that's really the best place to be. And certainly for me as a protein structure function person, you know, I got to play with and do way more proteins than I ever would have if I'd stayed in academia. You know, and then I mentioned before that I didn't want to do managing. I've had as many as 40 people in my group, but now leading the tech dev, I have 2. So if you looked at number of direct reports, you would think that my career peaked and then went down. But if you look at the influence within Amgen, it's definitely, you know, continued to grow. So I think I took a non-traditional career path, but it worked for me. So that's why I said I think it's important to know when to listen to yourself. But I would have stayed in the lab, perfectly happy doing the biophysics for I did a lot of the biophysics characterization. I was very happy doing that. And at one point, one of the VPs came to me and said, you know, we need somebody to lead this group who has kind of this strategy that you keep talking about strategy. If you're not willing to step up and do this, we're going to hire somebody from outside. And it scared me to be out of the lab. I actually haven't had a lab notebook for like 7 years, and that still bothers me. But you know, in terms of fun and influence and really getting to think about all sorts of different problems, it's been much better. But it did scare me really at first to do that too. So, and I think I mentioned I have kids. I have 2 kids who are 25 and 23 now. So, and in industry, actually, it's perfectly fine to say I planned my experiment, so I'm going to be gone from 3 to 7 because my kids are playing soccer, and then be online later. People don't actually care about the hours you work. All they care about is that you get done what you need to be done, and when you have to be there for an experiment, you are. And so I found for having kids, I can actually say I missed maybe only one of the important like games and things like that for my children. And I think that that's another thing. I have 2 of my the other woman who was in chemistry with me at Michigan went on to be a professor at Ohio State, and she can't say that. Now, partly that's probably our age, that it was harder, I think, back then. But I do think that that's another thing that industry can offer, is that people are less hung up on you putting in appearances at the right time, and really just care about you getting your job done. And if you're willing to work later at night because you want to do stuff with your family, that's perfectly fine. So,
Great.
anything else?
So I think we'll wrap up. Linda will stay here for a couple more minutes if you want to ask
Absolutely.
questions one-on-one. So let's thank her one more time for coming.